Hello, good morning to all of you. Today, in a line to better understanding of tumor microenvironment, I am excited to share some of the recent proposition that will facilitate today or in future various diagnostic as well as therapeutic avenues of cancer biology or cancer science. So what is the whole story today? We have to understand the tumor microenvironment that may be having relevance to the in vitro cell culture or in vivo model or ex vivo. In any case, whether cells or the tissue, in any particular tumor microenvironment, we are aware that within the tumor microenvironment, cancer cells are there, and within cancer cell, each cancer cells are unique or heterogeneous. They have their own genetic, epigenetic, metabolic identity. And other than cancer cell, there are non-cancer cells. Those are cancer associated cells, cancer supporting cell, that can be stromal cell, immune cell, and many more cells. And even nowadays, the idea of the microbiome, those are in the near vicinity of this tumor tissue. So altogether, whether cancer cell or non-cancer cell, including microbiome, it creates super complex environment. And that is why one of the proposition is coming up whether cancer cell and non-cancer cells, they share or exchange certain oncogenic resources. So the keywords today is to understand this story, that is oncogenic resources. These oncogenic resources, those are going to be exchanged between cancer cell to the non-cancer cell. And one more important point to this particular presentation is within cancer cells, and if we take the example now for chemotherapy treatment to the tumor, so majority of the cancer cells are going to be exposed and susceptible to the drug treatment. It can be classical like doxorubicin, cisplatin, or it can be targeted therapies. In any way, majority of the cells are going to be killed, and some of the cells are going to show the resistance, and they are going to survive, right? And once those cells are going to survive within a tumor tissue environment, so the next question is whether the cells or cancer cells, those are destroyed by the chemotherapy drugs, and those cells are dying cancer cells, they are going to go for apoptotic cell death or any type of the cell death mechanism, whether these group of the cells, those are destroyed by the chemotherapy drugs, whether they also share certain oncogenic resources to the surviving cancer cell. So this particular whole importance of understanding the nature of oncogenic resources, how the oncogenic resources are exchanged between the cancer cell to the non-cancer cell, and also within cancer cell, within the tumor environment during the chemotherapy treatment, how the dying cancer cells, those cells are about to go for cell death process, how they are going to exchange or share their resources at the time of their death process to the surviving cancer cell. And if we are able to understand those mechanism or the process, then we can also achieve certain success in terms of diagnostic and therapeutic avenues in cancer patients. So fine. So in that uh, before we will go for the proposed model, some burning questions are important. And we know that any drug treatment, any anti-cancer therapeutic options are not 100% efficient to kill all the cancer cells or even cancer supporting cells or cancer associated cells. So then what? The chance of relapse of cancer or the tumor is inevitable. It is going to happen because those limited number of cancer cells those are able to survive during this chemotherapy treatment or any other therapeutic treatment, they have already acquired certain oncogenic resources from the those cancer cells those were about to die or those cells are going to die. So that is one of the very critical aspect with reference to the tumor and the cancer. The second is why the dying cancer cell, the cells those are going to die, they will spare or they will, if you look at here, uh, these group of cancer cells, they will offer, they will spare their own important oncogenic resources because those cancer cells are about to die. So they do not need these resources, right? And in the next slide, we will discuss what are the nature of these oncogenic resources. Those are going to be exchanged between the dying cell to the surviving cells. And another very important is these oncogenic resources from cancer cells, those have already activated their cell death mechanism to the surviving cells, whether these are in the form of RNA, small RNAs, messenger RNAs, long non-coding RNAs, or metabolites like oncometabolites, or whole protein or peptide, and also to an extent extra chromosomal circular DNAs. And another very important question is relevant whether these molecules or biomolecules are offered and exchanged by these dying cancer cell to the uh, surviving cancer cell one by one, or it is exchanged through like hub-like mechanism where it is packaged within a particular structure like liposome structure, exosomal structure, and these complete hub of or packaged oncogenic resources are offered from the dying cancer cell to the surviving cancer cell. And that is why in the next slide we will have a proposed model, and then it will give a better clarity of this particular mechanism. Okay. Now, my dear friend, if you look at here, this is our tumor microenvironment. Okay. So the model is, this is the cancer cells, those are susceptible to the anti-cancer drug therapies, and the cells have started and ignited their cell death process, and the cells are going to die, right? So these cancer cells, those are about to die. So these cancer cells, one way they may offer, they may exchange, they may export it out from inside. This is the ICF, right? And this is ECF. So from the ICF of the cancer cells, these oncometabolites may be exchanged or they may be exported outside to the ECF. Look at here. Then, as I said, extra chromosomal DNAs, ECC DNAs. As we know, ECC DNAs are very important, and it has been already discovered the cancer cells have backup plan or the additional resources in the form of storage of oncogenes, and sometimes these ECC-DNA they code for certain oncogenic protein. So these ECC-DNA is a very important part of cancer cell growth and proliferation strategy. So whether these ECC-DNA is also going to be exported to the ECF as oncogenic resources, that is also very important to mention here. And then oncogenic small RNAs, microRNAs can also be exchanged or exported to the ECF from the ICF of this dying cancer cell. And the last question is, irrespective of ECC DNAs or small RNAs or oncometabolites, even large macromolecules like proteins, cytokines, even small peptides. These are also going to be exported to the ECF from the dying cancer cell. And all together, if you look at in a very holistic view, so these oncometabolites or ECCDNAs or small RNAs, proteins, cytokine, even complete messenger RNAs are being reported to be uh, exported to the ECF of the dying cancer cell. And all these molecules as well as the macromolecules are potentially exported as hub of oncogenic resources packaged within a large exosomal structure, the liposome structure, and then these hub of oncogenic molecule or the resources, once it reaches to the ECF of within a tumor microenvironment, then surviving cancer cells, those cancer cells have capability to escape out from the the ignited death mechanism of cancer cells. These surviving cancer cells potentially they are going to receive, look at, cancer cells may be received by these surviving cancer cell oncometabolites, exosome, ECCDNAs, or small RNAs, or many more macromolecule, including cytokine, protein, messenger RNAs. So what it gives us the idea is that the majority of the cancer cells those are going to die due to the insult by these chemotherapy drugs. At the end of or during the process of cell death, these cancer cells they are going to do a favor. They are helping their surviving cancer cell to achieve certain code, certain oncogenic code in the form of oncogenic resources, and so that these surviving cancer cells, even it is very limited in a particular tumor environment, these cancer cells have high power of resilience, relapse with so malignant potential. So the question is, these surviving cancer cells, they have the extra power of malignancies in the form of received oncogenic resources from this dying cancer cell to the surviving cancer cell. And it makes very logical thing, because whenever we say that cancer cells are going to die, it does not mean cancer cells are interested to die. Instead of, cancer cells are induced for certain cell death mechanism. It may be apoptotic or many more cell death pathway. But at the same time, these cancer cells, those are about to die, they do a favor. They create a microenvironment even at the time of the death process, so that it provides a seed or the platform for the next relapse of the tumor or resilience of the tumor. And that is one of the bottleneck in cancer field, or the for in the context of, if you think cancer patients, the relapse of the tumor is one of the very difficult situation. And that is possible because that is the whole intention of the tumor microenvironment, where even those cells are about to die. They create such oncogenic resources within the ECF, so that surviving cancer cell, they utilize those ECF, those oncogenic resources, for the future malignancies or future malignant potential. So what is the whole message here? The intention is, these chemical messengers, right? Whether oncometabolites or ECCDNAs or small RNAs, or you can say cytokine, messenger RNA, or long non-coding RNA, all that can be clubbed as oncogenic resources that has potential to transmit chemical messages from this dying cancer cell to the surviving cancer cell. And that may be one of the reason that the relapse or the resilience of the tumor is mostly observed in case of cancer patients. Based on this proposed model, then people are interested, what will be the translational values? How we can stop, to an extent, these exchange of oncogenic resources from a group of cells, those are going to adopt for cell death, and those cancer cells are going to survive within a tumor microenvironment. So for that, we need to go to the next slide. And here is so if we can develop certain approaches or therapeutic approaches, we can disrupt either export of oncogenic resources from the dying cancer cell to the ECF, and other way, if we can disrupt the import of oncogenic resources from the ECF to the surviving cancer cell. So the disruption of all these export and import mechanism of oncogenic resources can be a good therapeutic avenues for cancer patients. Also, this particular model may help us to go for diagnostic biomarkers. What nature of oncogenic resources, even at the time of the chemotherapy treatment or radiation therapies or many more therapies, what type of the oncogenic resources are generated within the ECF of tumor microenvironment is very, very essential, and that will lead to certain class of diagnostic biomarkers. And I shall appreciate if any question. Thank you very much. And best of luck.